here is your Unit 4 review, everything you need to know about homeostasis and membrane transport. So to begin, we will look at one single cell. The reason that I have it drawn like this is so that you can see just how many different solutes are in this, and this is ridiculously oversimplified. Most cells have thousands of different types of solutes in them. And cells also have all kinds of solutes around them in their environment. And that is the important idea of homeostasis, is that our cells are able to maintain a stable internal environment, keeping the solute concentrations, the temperature, the pH, and other conditions inside the cell different from the areas around the cell. And it doesn't matter what kind of organism you are, whether you are unicellular and only made of one cell, or multicellular and made of trillions of cells, the idea is still the same. You have to be able to keep each cell maintaining homeostasis. So here we have a single unicellular organism that has nutrients that are being used up. So these nutrients get used, and as they're used, they start to diffuse outward. Now, if you're a unicellular organism, you can easily exchange those materials with the environment. But if you're part of a multicellular organism, things get a little bit more complicated. You use up the materials, and so do all the cells around you. So then when those waste products diffuse out, pretty quickly you're left with toxic environment that you cannot sustain. So in multicellular organisms, that's where tissues and organ systems come into play, because that is going to enable you to move material into and through different parts of the body. The first organ system that we'll look at is the respiratory system, which is responsible for bringing oxygen into the body and removing carbon dioxide. Then we'll look at the digestive system, which will take glucose and other nutrients and bring them into the body. And then we'll look at the excretory system, which is going to be responsible for removing excess water as well as liquid waste materials out of the body. Throughout all of these, keep in mind that the cardiovascular or circulatory system is going to be the body system that's responsible for moving these materials from the respiratory system to the rest of the body, or from the digestive system outward to other cells in the body, or from other cells in the body down to the excretory system. So the cardiovascular system is going to have blood vessels that wrap around the respiratory system, wrap around the intestines and all throughout the digestive system, and is wrapping around the kidneys and going either to or from the heart, but regardless is going to act as a highway system to travel all throughout the body, right? So it's making sure that nutrients get everywhere from head to toe, so that nutrients are transported throughout the body in the blood, and then wastes are traveling in the blood out to the excretory system. And here we have a diagram of a typical capillary. That is a single cell thick layer, right? This outside of the capillary is just a whole bunch of cells that form like a tube. So the capillaries are the site of exchange of materials. So materials can move from certain tissues either into or back out of the capillaries to exchange those nutrients. And the blood plasma is going to be the liquid portion of the blood, whereas the red blood cells right in here are going to be one type of blood cell that we'll be taking a pretty extensive look at. And then finally, we also talked about two other body systems that are going to coordinate everything. The nervous system is going to be the brain, the spinal cord, and all of the nerves in the body, and that is going to be a very rapid but short-lived signal. And that nervous system is going to work in tight conjunction with the endocrine system, which is the body system that is responsible for making hormones. And there's a whole bunch of different endocrine glands throughout the entire body that is going to secrete hormones that are going to take a little bit longer to cause a response, but they are also going to be a little bit longer lived. So the response lasts longer in an endocrine response than in a nervous. So those are your 6 body systems that I would expect you to know. Now, when we're taking a look at the nervous and the endocrine system, both of these have a really huge role in homeostasis in that they're going to be able to detect any kind of deviation away from a set point as well as trigger some kind of response. So here's a typical negative feedback loop. Again, set point is going to be the ideal condition that any kind of change away from is going to lead to some kind of response that returns you back to that set point. The entire idea of a negative feedback loop is that if you deviate away from your set point, then you're going to have some kind of response that brings you back. Now, it's really important that you keep in mind the difference between the cause of the deviation or the cause of the stimulation of the stimulus, and then the response. The cause is what takes you out of homeostasis. The response is what brings you back. And let's take a quick look at what a typical negative feedback loop looks like. So something's going to happen that pulls you out of your set point or your set range. If we think of this in terms of temperature, any kind of drop in temperature, such as being exposed to cold air, etc., is going to be what lowers your set or lowers you away from your set point, and that's going to lead to your stimulus. The stimulus is any deviation away from the set point. That must then get sensed, usually by your nervous system, and some kind of response is initiated. So the sensor and the control center kind of work in coordination. And again, these are either the nervous system or the endocrine system typically, and that is going to send out a signal then to some other organ that is called the effector. Now that effector organ varies, depends on what the response is, but is going to do some kind of response to bring you back into homeostasis. And I think the easiest way to understand this is, as I said, kind of looking at temperature. So our set point is 98.6 degrees. We'll start by looking at what can cause temperature to drop. So the cause of a dropped temperature can be exposure to cold weather, for example. And that exposure to cold is going to change your body temperature in such a way that it reaches outside of your set range. So instead of 98.6 degrees, you have a colder temperature. And that drop in temperature in your blood is then going to be detected by a part of your brain called the hypothalamus. And your brain is then going to send signals out to a lot of different parts of your body to cause it to warm back up. So for example, you might start to vasoconstrict. And what that means is the blood vessels will become more narrow, and that limits how much blood, which is not only carrying liquids and nutrients, but it's also carrying a lot of heat. So it's going to reduce how much heat is brought to the surface of the skin and keep more of the heat in the body's core. Another effector that your brain is going to send signals to might be the muscles in the part of your skin, right under your skin, that is what makes goosebumps. And if you're really cold, the brain will signal to larger muscles in the body to start shivering. That shivering is going to cause ATP to be burned, and rather than use that ATP for energy, the chemical conversions of sugar into ATP and then the use of the ATP for various things is going to result in a whole bunch of heat that is produced. So the net effect is that when you are exposed to cold temperatures, or for some reason your body temperature drops, your brain recognizes it, sends a signal to a whole bunch of different organs to heat back up so that your body temperature rises back to the set point of 98.6 degrees. On the flip side, any kind of exposure to heat or any prolonged exercise is going to cause your body temperature to rise. And as we saw, when the temperature drops, the brain is also going to be responsible for sensing this rise in temperature. So we're going to have the brain then sending signals out to, just like before, the blood vessels, as well as some other organs. Now, in this case, the blood vessels are going to cause vasodilation, which is when the blood vessels open up really wide. That's going to allow more blood to get to the surface of the body, right under your skin. This is why you flush when you've been exercising a lot. And that vasodilation is going to allow more heat from the blood to get to the surface of the skin. It's also going to cause sweat glands to start releasing sweat, and that is going to, coupled with the vasodilation, allow more heat to escape the body because you will be sweating, and that is going to allow for evaporative cooling to occur. These activities are going to cause a drop in body temperature back down to your set point. So that's the basis of negative feedback loops. And in this case, we're looking at a two-way negative feedback loop, although that is not always the case. An important note here about fevers. When we talk about a failure to maintain homeostasis, what we mean is the body can no longer keep you at a set point. So I want you to think of this almost like if your thermostat in your house just stopped working. There would be no signal to the furnace when to turn on or when to turn off. So maybe it stays off for too long and your body temperature really drops. But in the case of a fever, homeostasis is working just fine. We've just changed the set point a little bit so that it's hotter, but not necessarily out of control, right? So we have our set point, and now we're setting it to, let's say, 102 degrees or so. We're still going to have the same negative feedback loop. So anytime the body is below now 102 rather than 98.6, if the body is below that, that triggers the response. So we have the stimulus of maybe your body's only 100 degrees or 101 degrees. That's still hotter than normal, but lower than the set point. So that recognizes it as too cold, and that's going to lead to eventually the effector of the muscles, and then you start to like shiver and warm up, and your body is stabilized usually at about 102, maybe a little bit warmer depending on what the new set point is. And as long as you have some type of infection, that set point stays increased, and that's usually actually healthy for you because that means you can fight off infections a lot better. Okay. We're going to switch gears and take a look at different types of membranes because you need to understand where membranes are found. Then we'll look at the structure of them. So what we have here is two different types of eukaryotic cells as well as the prokaryotic cell. We have an animal cell on the left and a plant cell on the right and a bacteria down at the bottom. Now all cells have a plasma membrane that surrounds them. So that's a layer of membrane that goes around the outside of the cell and separates the cytoplasm or the inside of the cell from its environment. So all cells, regardless, are going to have an outer plasma membrane. The difference between the prokaryotes and eukaryotes is in eukaryotes, we have membrane-bound organelles. In other words, we have membranes, the same kind of material that's found on the outside right here is going to surround various structures within the cell. And most notably, we're going to see the nucleus has a membrane-bound organelle, the nucleus is membrane bound. And then we're also going to have structures such as the endoplasmic reticulum. In this case, we've got the rough ER and the smooth ER. We're going to see that in plants and animals. We're also going to have the Golgi apparatus. So all of this is membrane. We're also going to have vesicles. Those are going to be responsible for transporting materials, and the organelles that are responsible for energy transformation, the mitochondria in the animal, and then in the plant we've got mitochondria and chloroplasts. So all of those things are wrapped in membranes, which means anything that they need has to go through a layer to be able to get into it, and any wastes that are produced have to go through a layer of membrane to get out. And this is what our membranes are looking like. So we've got a couple of key components. We'll start by looking at the phospholipid bilayer. And the phospholipid bilayer is exactly that. It is a bilayer, 2 layers of phospholipid, which is this structure right here. So the phospholipid is composed of 2 different parts, a phosphate head and 2 lipid tails. And what you'll notice is that in the phospholipid bilayer, the phospholipids are going to orient themselves so that the phosphate head, which is hydrophilic, it's water loving, it is polar, so it interacts well with water. The hydrophilic or phosphate heads are pointing up and down, so they form the outside layer of the phospholipid bilayer. And the reason that they do this is that the entire inside and outside of the cell is primarily made out of water. It's an aqueous environment, and so if we've got water on one side of the membrane and water on the other, the only orientation that's going to allow for the lipid tails to stay tucked away is one in which the hydrophilic heads are facing the water. And that leaves the hydrophobic tails facing in the middle, kind of sandwiched in between this layer. So we've got this core of the membrane that is hydrophobic, and then this outside that is hydrophilic. In addition, we also have cholesterol molecules. And cholesterol is a type of lipid, which means that they are going to be hydrophobic as well. So they dissolve really easily in the lipid tails because both of those things are hydrophobic, and like dissolves like. In other words, things that are hydrophobic interact with other things that are hydrophobic. We've also got these guys right here, which are various types of proteins. And as you remember, proteins are made out of different amino acids. So some amino acids are polar, some are nonpolar, and the protein is going to arrange itself in a way that any part of the protein itself that is polar is facing out either into the outside of the cell or into the inside of the cell, while any part that is nonpolar kind of rests here in the middle. And the proteins are going to be really important to work as channels that allow things through, as well as pumps and a whole bunch of other functions. And then finally, there's some other stuff, and that's all you really need to know, is that it's other stuff that is embedded in the membrane and can be really important for cell signaling, saying, hey, like I'm a cell that can respond to insulin, for example. It's also going to be really important for anchoring cells together and various types of communication. But again, don't worry so much about that. Your main focus is making sure that you know what the phospholipid bilayer is, so 2 layers of phospholipid here, that proteins are going to be embedded all throughout this phospholipid. In fact, proteins and phospholipids make up the majority of the cell membrane. And then we also have some cholesterol here that's helping with maintaining the integrity of the phospholipid bilayer, as well as keeping it nice and fluid. Okay, so let's talk about molecular movement here for a minute, and we'll start by looking at this idea of a concentration gradient. A concentration gradient is literally just that, a gradient or difference in concentration from one area to another. So over here you can see we have a low concentration of whatever this blue solute is, and over here we have a high concentration. And what molecules do is they're in completely random motion. Any one individual molecule or particle is going haywire all over the place. And the net effect of this is that overall you tend to have a movement of molecules from an area of higher concentration to lower concentration. And this is called diffusion. And diffusion is going to cause molecules to disperse until we reach something called equilibrium. Equilibrium means just that everything is spread out. It does not mean that they're not moving. It just means they're not, there's no net movement. So overall, this individual molecule might be over here, it might go over here, it might go over there, but the net effect is that they're pretty well spread out throughout the entire area. So now let's say you have a membrane that is separating an area of higher concentration to lower concentration. If that membrane is permeable, meaning that it's going to allow things through, so if it is permeable to the various substances, or in other words, the substance can move its way through, then you will end up via passive transport having equilibrium reached, where both sides of the membrane are the same. Okay, if the membrane were permeable to everything, then the cell would be in complete equilibrium with its environment, and that is the exact opposite of what we're looking to be able to go for with homeostasis. So for the next few slides, we're really looking at finding a way to make a high concentration in one area of whatever particular solute is, have it cross the membrane because the membrane is permeable to whatever that substance is, and then we can get it into the cell or out of the cell. And we'll start that discussion with the respiratory system. The respiratory system is composed of a number of organs that you should at this point be pretty familiar with, starting with the nose and the mouth here. We've got the trachea, or the windpipe, that goes down. This is going to separate into separate bronchioles, which are larger tubes, into smaller and smaller tubes successively, until finally we end up in these little air sacs, these kind of end of the road pieces down here that kind of look like grapes. Now let's take a closer look at exactly what those look like. Each of these air sacs, there we go. So we've got the tubes right here that kind of veer off to make these various air sacs, right? And they're kind of coming in all directions. And you notice all the blood vessels that are surrounding it, and that's because we're going to have oxygen that is coming in. What's a good color for oxygen? Okay, whatever. My bad. There we go. So we've got oxygen coming down these tubes, up to the ends, and into the air sacs. And then once the oxygen gets to the air sac, the oxygen is at the highest concentration that we're going to have in the entire body, and then it is going to diffuse or move via passive transport to get into the capillaries. So that's going to look something like this. So we've got a really high concentration of oxygen on the air sac side, and a lower concentration on the in the capillaries. And we're cutting out a couple membranes here. That's okay. The air sacs are wrapped in cells, so each one of those is going to be 2 cells thick, and so on. Anyway, but oxygen is a small nonpolar molecule, and that means that it is going to move directly across the cell membrane via simple diffusion. Okay, anything small and nonpolar is going to be able to do this. So that is how our respiratory system is going to be able to move material from the environment down into the lungs and eventually into the blood. The converse is going to be true in terms of getting carbon dioxide out. We're going to have the highest concentration of carbon dioxide in the capillaries here. So that's going to then diffuse into the air sacs and be expelled each time we exhale. And so let's talk about how the respiratory system maintains homeostasis. We have a set point that is, we're just going to keep this easy, enough oxygen to be able to work. Okay, keep that easy. Now, what's going to cause a change to the set point is this process right here, which we've talked about ad nauseum, right? We're taking glucose, which is sugar, and we need oxygen. So we're going to be using that oxygen to break down the sugar or the glucose, and that's going to produce carbon dioxide. So what we need is a high concentration of oxygen. However, if we do things that are requiring a lot of ATP, then this entire process is going to happen a lot, and we're going to start using up the oxygen that we have in our body and causing those rates to drop. At the same time, we're also going to be producing a whole bunch of carbon dioxide, so those amounts are going to increase. So we have a decrease in the oxygen concentration and an increase in the carbon dioxide concentration anytime we do something that requires a lot of ATP. So what's going to cause a change to the set point of oxygen, right? What's going to cause a change in that set point is exercise. Boom. Or not breathing enough, or really anything that's decreasing your oxygen, but exercise is going to decrease oxygen most commonly. Now, as a side effect of all of this is we've got our carbon dioxide concentrations that are starting to go up. We've got water all throughout all of our blood, and that means that these two, the carbon dioxide and the water, are eligible to perform a nice little chemical reaction that produces carbonic acid. So keep that in mind. In fact, that is what our stimulus is. It's not that the oxygen levels are down. It's not that the carbon dioxide levels are up. What the brain actually detects, our stimulus here, is that the blood becomes slightly acidic. In other words, the blood pH drops because we remember that low numbers are more acidic. Okay, so we can now, understanding that, go back to our set point. Having enough oxygen means that our pH is roughly 7.3, 7.4-ish. Okay, when we exercise, we use up oxygen, we produce carbon dioxide. The carbon dioxide produces carbonic acid. Carbonic acid is an acid, you're welcome, and that's going to cause the blood pH levels to drop, which indicates a drop in oxygen and an increase in CO2. Easy, right? Sure. Okay. In this case, just as we've already seen, our sensor and our control is the brain. You don't need to memorize the particular parts of them. Just know that the brain is like, yoinks, this is a problem. And the brain is going to send signals to the diaphragm, which is just under your lungs. And really, it all comes down to the respiratory and the cardiovascular system. So the brain says, we don't have enough oxygen. So what are you going to do to bring it back? How does the effector return the condition to set point? This is easy. Think about this one through. If you exercise and you have low oxygen, how do you get more? You breathe faster. What? Crazy. You know this. Just make sure to think it through. Okay. What's really great is that on the flip side, we don't need anything over here. If the pH level starts to increase, meaning go above the set point, become more alkaline. For example, if you're like hyperventilating and you can't get your thoughts straight and you're breathing really fast, what's going to happen? Well, your blood starts to get a little bit alkaline, but that's okay because then, there we go, because then the stimulus of the drop in pH goes away. And so no stimulus, boom, boom. The brain stops telling the respiratory and cardiovascular system to breathe faster, and the net effect is that you're going to breathe slower and it kind of self-regulates. Okay? Okay, so now we're going to take a look at the digestive system. This is where sugars and other nutrients are brought in, and hopefully you're pretty familiar with this guy too. So we've got the mouth, we've got the esophagus. I want to point out something here. Remember that PHAG, that phag? We're going to be seeing that later in bulk transport. It means to eat. So guess what your esophagus does? It transports food. And where does it go to? It's going to go down the esophagus and then into the stomach, which is right here. The stomach's really going to be where digestion starts to occur, but what we care the most about is absorption. So once we've got the nutrients broken down and digested in the stomach, they're making their way to the small intestines. Now, glucose does not diffuse directly through the phospholipid bilayer. Instead, glucose is going to need channels, and that is going to be via facilitated diffusion that we see glucose kind of moving from the inside of the small intestine through a whole bunch of layers of membranes and different cells until finally it gets into the cells and into the blood. That's going to then allow the glucose to be carried. So what is it doing? Well, these glucose molecules, as I said, it's too big, they're too polar, and they cannot get directly through the membrane. Instead, what they're going to be doing is going through channels, and cells have to have channels to allow each particular item through. In this case, the channel is a glucose transporter or a glucose channel. Every cell happens to have glucose channels because, well, glucose is kind of darn important. So there we go. In terms of regulating your homeostasis, you have two things that can happen. You can have a drop in blood sugar or a raise in blood sugar. Your normal level is about 80 to 100 milligrams of sugar per deciliter of blood. That's the glucose test. If you ever have that done, you want a number between about 80 and 100. Okay, so think about this logically. What causes your blood glucose levels to drop or to raise? Well, what would cause you to not have very much sugar in your body would be not eating a lot of sugar. And anytime you burn sugar, such as when you do cellular respiration, that is also going to cause a decrease to the overall amount of sugar in your body. And both of these conditions are completely normal. You do them all day, every day. Your body's very, very used to having a little bit of a drop towards the lower end of your set point, and it just responds and gets you back to where you need to be without too much fuss for most of us. So you've been fasting, for example, overnight, or you've been exercising, and the amount of sugar in your blood drops. And instead of your brain being what detects that change, instead we're going to have an organ that's kind of located in the tuck right here of where the stomach meets the small intestine. And what that looks like, we can come back over here. And what we have is an organ right in here. It's kind of a fluffy little yellow guy called the pancreas. And the pancreas is going to be what is in charge of recognizing and responding to any kind of change in blood glucose levels. So we have our pancreas, and that is going to send a signal to a bunch of different parts of your body. And to understand the response fully, we're going to take a quick little peek over here and actually look at what's happening and on the flip side. When you eat, that is going to cause your blood sugar levels to rise. So you have more blood sugar. And again, just as we saw when your blood sugar levels drop, the pancreas is going to be in control of making the response. So it senses it and then sends out the signal. In this case, our signal is going to be the release of something called insulin, which is a hormone. And insulin is going to be sent throughout the entire body, but there's only certain cells that are going to be able to respond to that. So that is going to include the liver, the muscles, and the fat cells. What these various tissues are going to be able to do is when they sense insulin is present, they're going to take the extra sugar in the blood and convert it to glycogen. So just a quick review here. Glucose is a monomer of carbohydrates. It is the monosaccharide that when you polymerize it or put it into long chains, you end up with glycogen. So that is our storage molecule. So insulin is the hormone that tells the liver, the muscles, the fat cells to take up that glucose and convert it into glycogen. And in fact, now that we've kind of finished up the unit, maybe you can appreciate this a little bit more. Glucose is much, much smaller, and glycogen is really, really big. So glucose is going to contribute a lot more to the molarity of the blood plasma, whereas glycogen is not going to have so much of an effect. Well, when you have a lot of glucose, that's a lot of solute that you have in your blood, in the plasma, in the liquid part of your blood. So hopefully you're thinking something about osmosis. I'll come back to that in a little bit, but I just think it's a good connection to make. Okay, so why did I mention that? Well, we need to understand that the body is storing extra sugar as glycogen. So when your blood sugar goes up, you take the extra, you store it as glycogen, and that's going to drop the sugar levels back down to normal. Okay, so on the flip side then, when your blood sugar levels drop, your pancreas can recognize this, and it is going to secrete a hormone called glucagon. And in this case, the cells that respond to the presence of glucagon are mainly going to be the ones in the liver. And the liver is already going to be full of glycogen, and now the reverse is going to be true. We're going to take the glycogen, and when this signal comes in to do so, the liver is going to take that glycogen and convert it down into glucose. So glucagon is that signal. Glucagon is the hormone that's telling the liver to convert glycogen back into glucose. The result is that now the liver cells have an increased amount of glucose in them, and that can then diffuse out via facilitated diffusion back into the blood and raise your blood glucose levels back to normal. Our final topic here in terms of the body systems are going to be that of the excretory system. And the main organs of the excretory system are the kidneys and the bladder. And I like this diagram a lot because it really shows you just how important blood sources are. So here we go. This is your descending aorta. So the main artery that goes from the heart down to the lower part of the body. You can see what a huge artery is going to the kidneys. And then in terms of blood return, this huge vein right here, these sets, are going right back up to the heart. So there's a ton of blood flow in the kidneys. And the reason for that is because the kidneys are what are going to be filtering out all kinds of stuff from the blood and either sending that to the bladder or putting that back into the blood. And the excretory system is going to work primarily off of a system called osmosis. And osmosis works by having aquaporins, which are specialized protein channels that allow water through. So this is a form of facilitated diffusion, but it's a little bit different in that instead of having the solute go across, you're now having the solvent go across, so water. We have to be able to describe the two sides of the membrane when it comes to osmosis. An important thing to note is that the terms hypertonic and hypotonic only are applicable when we're talking about osmosis. So the side with a higher solute concentration is hypertonic, meaning in this case it's got more of whatever these little triangle doodads are. And importantly, these triangle doodads, these solutes, cannot have another way of going through the membrane. If the membrane is permeable to the solute, then it does not contribute to tonicity. So the hypertonic side is the side that has more solute that is not permeable, whereas the hypotonic side is the side with less, and consequentially, it is also going to be the side that has a relatively higher amount of water molecules. So more water on the hypotonic side and more solute on the hypertonic side. Osmosis is always going to take water from where there's a higher amount of water, a lower amount of solute. So from the side that is hypotonic to the side that is hypertonic. And that's going to go through the aquaporins there. In terms of regulating, well, why would you need to? Well, when your blood plasma and your blood cells and all the other cells in your body are isotonic, that means you've got the same amount of water coming in as going out, and your cells are happy. They're working great, and everything is right where it needs to be. However, if your blood gets really, really hypertonic, meaning the area around the blood, the liquid portion, the plasma, if that gets hypertonic, then water is going to be moving out of cells and into the plasma, right? So from cells to the liquid to that surrounding area. So blood equals hypertonic, okay? So what would cause your blood to become really hypertonic? Well, anything that increases tonicity would do that. So any kind of intake of salt or anything that decreases the amount of water that you have in your body. For example, if you're exercising a lot, you'll be sweating out a lot of water. That water loss means that you have less water in your blood, and so your blood becomes hypertonic. Also, vomiting and diarrhea. There's a lot of illnesses out there that the illness themselves are not the danger. It's how much water you lose during the course of that infection. That's the real danger. So if you have a lot of salty foods, or you're sweating a lot, or you are vomiting and have diarrhea, or you're just not drinking enough water, then your blood is going to become hypertonic. And in this case, that is going to be sensed by the brain. The brain is going to send signals to the kidney, and the signal that they send is a hormone. So we're back to thinking about the endocrine system, called antidiuretic hormone. An antidiuretic hormone literally means the no pee hormone, because a diuretic is anything that causes you to urinate. And so antidiuretic is something that tells you not to urinate. So the brain is going to cause a release of ADH, and that is going to be recognized by the kidney. What the kidneys are then going to do is collect water, just like they always do, but then the water is returned to the blood. So that's going to really help us conserve as much water as possible when we're dehydrated. On the flip side of that, anything that's causing your blood to become really hypotonic could potentially cause the blood cells to burst, and can also cause a lot of swelling in the brain and other organs as water is getting infused into those cells. So hypotonic blood is not always a very good thing. In fact, if it's too hypotonic, that's really bad. However, What makes you hypotonic most of the time is just drinking water. It'll make your body a little bit hypotonic, but so long as you can maintain homeostasis, you regulate it and life is good. So just like before, we're going to have the blood is recognized as being hypotonic by the sensor. In this case is the brain. That is going to then stop sending ADH. And the kidneys recognize this drop in ADH and they respond by, instead of filtering out the water and returning it to the blood, they're going to collect out the water and send it to the bladder. By sending extra water to the bladder, you reduce how much water you have in your blood. Your blood returns back to being isotonic with the cells in them, and everybody is happy. That's the ugliest smiley face ever. That's okay. All right, so quick recap, and something really important that I want to point out because I've seen a lot of people make this mistake. Cause, effect. These are the things that you're doing down here, right? You're eating salty food, you're sweating, you're vomiting, or you have diarrhea. The things that are pulling you away from homeostasis. All of the rest of this is the effort that your body makes to get back into homeostasis. So if you have too little water, your body's going to do things to get more water or at least save what it has. If you have too much water, your body's going to get rid of the extra. Please, please, please keep in mind cause and effect. Make sure to study. And if you've got questions, let me know. I did not cover active transport. That is in a separate video. And again, if you've got questions, I got your back. You just let me know.